This video is for The Living Earth, Unit 4.1, Genetics. Today's essential question is, what are the effects of multiple alleles, codominance, and incomplete dominance on phenotype? Multiple alleles.
Human blood type, or blood group as it's sometimes called, is a classification system that is based on antigens present on the surface of red blood cells. Although there are over 30 recognized human blood groups, the ABO blood group system is the most important classification when blood transfusions are concerned. The ABO blood group system was discovered by Austrian scientist Karl Landsteiner in 1900. Landsteiner discovered the three blood types, A, B, and O, and shortly afterwards, in 1902, Alfred von de Castello and Adrian Stirley discovered the fourth type, AB. The discovery of these blood groups led to much safer blood transfusions. You see, when the blood from two individuals with different blood types is mixed, the recipient's blood cells may contain antibodies against the donor's blood cells, which can lead to blood clumping, cells cracking, and toxic reactions, which can have fatal consequences. Blood type is inherited from our parents, and one gene is responsible for the ABO blood group system producing four possible phenotypes for human blood: A, B, AB, and O. We know that with incomplete dominance and codominance, two alleles of a gene can result in three possible phenotypes. But what's happening with human blood? How can one gene produce four phenotypes? If four or more phenotypes are expressed by a particular gene, then there must be more than two alleles that exist for that gene. We call this multiple alleles. It's important to understand that individuals can only have two alleles for a particular gene, one that they inherited from their mother and one from their father. But although individuals can only have two alleles, there may be three or more alleles that exist within the population. Human blood type is a classic example of multiple alleles. The ABO gene has three alleles: uppercase I type A, which codes for type A blood, uppercase I type B, which codes for type B blood, and lowercase I, which codes for type O blood. Thus, we can see that these three alleles have six possible genotypes, and these six genotypes result in the four different phenotypes which represent the four blood types: A, B, AB, and O. Did you notice that lowercase I, or type O blood, is recessive to uppercase I type A and uppercase I type B? Did you also notice that uppercase I type A and uppercase I type B are codominant? Let's take a look at a couple of examples of this using a Punnett square. In this example, we cross a person with type AB blood with a person who is heterozygous for type A blood. From the Punnett square, we see that four distinct genotypes are produced: AA, AB, AO, and BO. However, the resulting phenotypes are 50% type A, 25% type AB, and 25% type B. In this next example, let's determine if a man with type B blood and a woman with type AB can produce a child with type O blood. Since type B blood can exist as two different genotypes, we have to look at two different Punnett squares to represent this scenario. Type O blood can only come from the genotype lowercase I, and by working both of these Punnett squares, we can see that none of the combinations result in lowercase I. Thus, the answer to this question is no. Blood type is just one of many examples of genes that have more than two alleles in a population. In humans, eye color, eye shape, hair color, hair texture, and our physical body structure are all examples of traits that are determined by multiple alleles.
Codominance.
Combinations of alleles at a locus for a particular gene produce various phenotypes in organisms. Dominance is the relationship between these allele combinations with respect to the phenotypic outcome of these combinations. Complete dominance occurs when the phenotype of the heterozygote allele is the same as the dominant homozygote allele, because the dominant allele completely masks the effect of the recessive allele. For example, a pea plant flower has a dominant allele, R, which produces a red color, and a recessive allele, R, that produces white color. Flowers with the RR or RR allele combinations will both produce red flowers, while RR combinations produce white flowers. Incomplete dominance occurs when the heterozygous combination produces a phenotype that is distinct or intermediate from the homozygous genotypes. For example, when a red flower and a yellow flower produce flowers that are orange. So what about a situation like this, where a red-brown cow and a white cow produce offspring that are spotted white and red-brown? Before we answer that, let's review how we inherit our traits from our parents. For every gene, we have two alleles, one of which we receive from our mother and the other from our father. As we mature, we produce gametes, our egg or sperm, which pass these traits on to our offspring. One of the ways in which we can predict the probability of the genetic outcome of breeding is to use a Punnett square. If we look at a monohybrid cross, where we're only examining the combination of one pair of alleles from both parents, the Punnett square allows us to see what are all the possible genotypes that can be produced. Let's take a look at the case for the color of a rabbit's coat and set it up with a Punnett square. The B allele produces a black coat, and the B allele produces a white coat. Let's use a scenario where both parents have the genotype BB. When we work the combinations using the Punnett square, we can see what genotypes can be present in the offspring. In this example, we can see the probability of the offspring having a genotype of BB is 25%, BB is 50%, and BB is 25%. It is important to note that the Punnett square only provides us with a probability of genotypes, not the resulting phenotypes of the offspring. Remember, phenotypes are a result of how different genotypes are expressed. In our pea plant flower example from earlier, the heterozygote produced red flowers in the complete dominance scenario. Codominance occurs when the phenotypes of both alleles are expressed as a heterozygote, meaning both phenotypes are dominant. Human blood type AB, which is comprised of both blood type A allele and blood type B allele, is a classic example of codominance. Going back to our spotted white and red-brown cow, we can now see that this is a result of codominance. Spotted cows are heterozygous for coat color and are codominant. Therefore, both colors are expressed and appear on the coat. If we set up a Punnett square for a cross between a homozygous white cow and a homozygous red-brown cow, we can see that all the offspring would be spotted. What if two spotted cows were crossed? Here, we can see that the offspring's probability would be 25% red-brown, 25% white, and 50% spotted. Here are just a few other examples of codominance in the natural world. Non-patterned black or brown homozygous cats can produce offspring that are patterned. And erminette chickens are another example where homozygous black or white parents can produce offspring that have mixed feathers of black and white.
Continuing with incomplete dominance.
Dominance is the relationship between allele combinations at a locus for a particular gene and the resulting phenotype in that organism. Let's say a flower has a dominant allele, R, which produces a red color, and a recessive allele, R, which produces a white color. Flowers that have the RR allele combination will produce red flowers, and flowers with the RR combination produce white flowers. However, flowers that have the heterozygous allele combination RR will produce red flowers, because R, or red, is dominant to R, or white. Here are just a few examples in humans of dominant and recessive traits. Hanging earlobes are dominant to attached ones. Being able to roll your tongue is a dominant trait to not being able to. And although eye color varies widely, blue eyes are typically recessive to non-blue. But what happens in a situation like this, when a red flower and a white flower produce an offspring flower that is pink? Or how about a black cat and a white cat producing gray kittens? Or one parent having straight hair, the other curly, but the child has wavy hair. All three of these situations are examples of incomplete dominance. This occurs when the heterozygous combination produces a phenotype that is distinct or intermediate from the homozygous genotypes, RR. Using the pea plant flowers example from earlier, in a normal dominant scenario, the RR combination produces a red flower, because red is dominant over white. However, in an incomplete dominant scenario, the combination produces an entirely unique phenotype, because one allele is not completely dominant over the other allele. Therefore, RR would produce red flowers, WW produces white flowers, and RW produces pink flowers. Incomplete dominance is also at work in the case of the gray kitten and the child with wavy hair. The genes for these traits exhibit incomplete dominance, and these offspring, like the pink flower, are also heterozygous allele combinations that have an entirely unique phenotype. One of the ways in which we can predict the probability of the genetic outcome of breeding is to use a Punnett square. If we look at a monohybrid cross, where we're only examining the combination of one pair of alleles from both parents, the Punnett square allows us to see what are all the possible genotypes that can be produced. For instance, let's take our cactus flower example from earlier. Let's assume the pink flower has the RW combination and the white flower has the WW combination. Using the Punnett square, we can see that the probability of the offspring having a genotype of RW or pink is 50% and WW or white is 50%. If a red flower had an RR combination and was crossed with a white flower with a WW combination, the Punnett square shows us that 100% or all of the offspring would be pink flowers. If two pink flowers were crossed, we can see that the probability of the offspring being RR, which is red, is 25%, RW, which is pink, is 50%, and WW, which is white, is 25%. Here are just a few other examples of incomplete dominance in the natural world. The combination of black and white homozygous sheep produces gray sheep and heterozygous offspring. Andalusian fowls are another example where homozygous black and white parents can produce heterozygous offspring that are blue. In humans, sickle cell anemia is an example of incomplete dominance. So let's review. The difference between dominance and incomplete dominance is the phenotype of the heterozygous allele combinations. In dominance, the heterozygote takes on the phenotype of the dominant allele. In incomplete dominance, a completely unique phenotype is produced that is different from both the dominant and recessive alleles.
Sex determination. Most organisms that reproduce sexually come in two forms, male and female. The chances of a child being born a boy or a girl are about 50%, or 1 to 1. But what determines the sex of a child? Human cells contain 23 pairs of chromosomes. In females, the chromosomes in each pair look identical, but in males, 22 are similar, but one pair is different. In males, this 23rd pair consists of one large chromosome, which we call an X chromosome, and one small chromosome, which we call the Y chromosome. In females, the 23rd pair consists of two similar XX chromosomes. These chromosomes are called sex chromosomes. During meiosis, males produce sperm that contain either an X chromosome or a Y chromosome. Females produce eggs containing X chromosomes. When a sperm fertilizes an egg, the probability of the zygote containing two X chromosomes is 50%. One X from the mother and one X from the father. The chance of it containing an X and Y chromosome is also 50%, the X from the mother and the Y from the father. There is therefore an equal probability of the child that ends up developing either a boy or girl. Polygenic inheritance. When Mendel selected traits to study in his pea plants, he chose ones that, unknown to him, were controlled by single genes. This greatly simplified his work, but does not give a complete picture as to how most phenotypes are determined. Most phenotypes are controlled by more than one gene. Because of this, they are referred to as being controlled by multiple genes, or polygenic. These polygenic traits, such as height, hair color, or skin color, show continuous variation. For this reason, they do not follow the simple phenotypic ratios of Mendelian inheritance. For example, human skin color depends upon the amount of melanin, which is a dark skin pigment, that it contains. The genetics of skin color are quite complex, but it goes something like this. A number of genes control the manufacture of melanin in the skin. For each of those genes, there are two possible alleles. One allele that contributes to melanin production, and one allele for non-melanin production. The more alleles for melanin production that you have, the darker your skin. Parents will have different combinations of these alleles, which they will then contribute randomly to their offspring. The skin color of a child depends upon which alleles it gets from each parent. Their skin may well be different shades than either parent. Height is another example of polygenic inheritance. People are not just tall or short, they show a continuous variation in height. Scientists have identified over 400 genes that may be involved in determining height. These genes contribute to the many processes that determine height, from cell growth to bone shape. Of course, these multiple genes are not the only factor that determines height. The environment, particularly diet, also plays a part. At this point, you should be able to generate a full and appropriate response to today's essential question. What are the effects of multiple alleles, codominance, and incomplete dominance on phenotype?